In this image, B cell development begins in the bone marrow from pluripotent hematopoietic stem cells. These stem cells differentiate into pro-B cells and then pre-B cells, during which immunoglobulin gene rearrangement occurs. The B cell lineage marker CD19 first appears at the pro-B cell stage and continues to be expressed throughout most stages of B cell development. As the cell matures into immature B cells, they begin to express surface immunoglobulin and acquire CD20 expression. These immature B cells then leave the bone marrow and enter the peripheral circulation as naive mature B cells. In the peripheral lymphoid tissue such as lymph nodes and spleen, naive B cell encounter antigens and become activated. The activated B cell enter the germinal centers where they undergo clonal expansion, somatic hypermutation, affinity maturation, and class switch recombination. These processes enable the production of high affinity antibodies and the selection of effective B cell clones. Throughout these stages, both CD19 and CD20 are expressed on B cell surface. Following the germinal center reaction, B cell differentiate into either memory B cell or antibody secreting plasmablast. Memory B cells are long-lived cells that provide rapid and enhanced immune responses upon re-exposure to the same antigen. Plasmablasts represent an intermediate stage that actively secretes antibodies and eventually mature into plasma cells. During this transition, CD20 expression is lost, whereas CD19 may still be present on some plasmablast. Plasma cells are terminally differentiated B cells that produce large amount of antibodies and primarily reside in the bone marrow and lymphoid tissue. Most plasma cells don't express CD20 and may lose CD19 expression as they fully mature. This pattern of marker expression has important therapeutic implication. Anti-CD20 therapies such as rituximab effectively deplete immature, mature, activated and memory B cells but do not directly eliminate plasma cells, which explains why existing antibodies can persist even after B cell depletion. In summary, CD19 is a broad B cell lineage marker that appears early at the pro-B cell stage and persists through most stages of development, whereas CD20 appears later at the immature B cell stage and is lost when B cell differentiate into plasmablast and plasma cells. Understanding this developmental pathway is essential for interpreting B cell immunology and mechanism of action of B cell targeted therapies used in autoimmune diseases, neurological disorders, and hematological malignancies.